Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and we are into the revision notes, revision mode, and we have been revising chapter wise. In our previous session we revised chapter 3 plant kingdom, and in this session we are going to revise the syllabus and important points of chapter number 4 animal kingdom. So basically this chapter deals with the classification of animals into phyla and what are the various reasons for classifying animals into various phyla or groups. What is the logic that we apply when we keep a particular animal in a particular group, and where is the necessity for all this? Is there really necessity for this? Yes, of course, all of us accept that there is necessity because there will be a lot of confusion if there is no classification. Just like in plant kingdom, how we classified animals based on the characteristics, similarities, dissimilarities, evolutionary features, similar pattern we adapt in this chapter also. So with that background, moving ahead, millions of species of animals have been described and it becomes more necessary to classify them to assign systematic position. Many times in your own native place, you must have seen in your own field some animals, and after observing the morphology of the animal, external features of the animal, being a biology student, still you wonder what is that animal, what is the name of that animal, to which phylum it belongs, to which group it belongs. A lot of discussion will be there. Sometimes you are disappointed that you are not able to identify. So to overcome such situations, we need this classification. So basically, animals are classified on the basis of arrangement of cells. First of all, whether cells are there, cells joined together, tissues are formed, and whether tissues form organs, organs joined together, organ systems are there. So this is the first thing. Then body symmetry. What is the shape of the body of the animal? Then nature of the coelom. Coelom is body cavity, guhe present inside the animal. Whether coelom is present, true coelom is present, or coelom is not at all present, based on that. And then pattern of digestive, circulatory and reproductive systems. So these are the general characters we take based on which we classify the animals. So first one, levels of organization. Cellular level, sponges, example. Tissue level, cnidarians, okay, and we call it as coelenterata. Previously old name is coelenterata and new name is cnidarian. Organ levels, of course, platyhelminthes, flatworms. Organ systems, arthropoda, cockroach family. So incomplete digestive system has one opening, but complete digestive system has two openings, mouth and anus. Unfortunately, there are many animals whose digestive system has got only one opening. That means both intake and outgo is through the same oral aperture. How bad it is. And then some animals have open circulatory system. That means blood will not be pumped out of heart, and cells and tissues are directly bathed in it. That means there are no, what we are having, arteries and veins. Blood will not flow through the tubes, artery and veins. Instead, just like a swimming pool, like you must have observed in cockroach. And closed circulatory system, blood is circulated through, as I said, arteries, veins and capillaries. So second base on which we classify the animals is symmetry, shape of the animal. Some will be symmetrical, can be divided into two exact halves, then we say bilateral symmetry. Asymmetrical, you divide them, cut them into any in angle, you don't get two equal parts, then we say asymmetrical, shapeless fellows like amoeba, sponges, they don't have specific shape. And once animals are symmetrical, whether when we cut the animal in a particular axis you get two exact halves, then we say bilateral symmetry, can be divided into equal halves by only one plane. Example, all vertebrates including us. Whereas radial symmetry can be divided into equal halves by any plane passing through center, like starfish, hydra. This is radial symmetry. Of course, when we talk of the hydra, this is the embryo form. The animals in which cells are arranged in two embryonic layer, external ectoderm, internal endoderm, is called diploblastic. So during the embryonic stage, if an animal has got two layers within the embryo, external ectoderm, internal endoderm, then we say diploblastic animal. Example, porifera, coelenterata or cnidaria, like this. When you cut open this embryo, we can see the presence of two layers like ectoderm and endoderm. This is ectoderm, this is endoderm. Then we say bilateral. And if we see three, then we say two layers if you see diploblastic. Three layers if you see triploblastic. That is ectoderm, mesoderm, in between, middle. Meso means middle, derm is layer. Endo means inside, derm is layer. Then we say triploblastic animal. So the animals in which developing embryo has a third germinal layer, mesoderm, besides ectoderm and endoderm, then such animals are called triploblastic animals. Example, platyhelminthes, chordates. And next basis for classification is body cavity. Body cavity is called coelom. This is the coelom. This is coelom, okay, in the inner space. The body cavity which is lined by mesoderm is called coelom. Animals possessing coelom are called coelomates. Like all of us, annelida, chordates, mollusca, these are coelomates. That means you got ectoderm, endoderm and mesoderm, triploblastic, and then coelomates. Inside there is one cavity. That body cavity is coelom. In some animals, cavity is not lined by mesoderm but scattered as pouches. As you are seeing here, see, this is one bag, this is one bag, this is one bag, this is one bag. So this is not lined with the mesoderm. Instead, scattered as pouches between ectoderm and endoderm. Then we say pseudocoelomates. Example, ask helminthes. The animals in which body cavity is absent, there is no coelom at all, then acoelomate animals, platyhelminthes. As the name says, flat. There is no need for coelom, acoelomates. So you got, based on the presence or absence of body cavity, if coelom is there, coelomates. If coelom is not there, acoelomates. Coelom appears to be there in the form of pouches, then pseudocoelomate. In some animals, body is externally and internally divided into segments with serial repetition, as an earthworm. Then we say metameric segmentation. Body is divided horizontally into steps. And with all these bases, we classify the animals into different phyla. So we start up with first one, phylum Porifera. Members of this are commonly known as sponges. You know, when you are sitting on your sofa set, on your chair, it will be very, very soft. That softness is because of the sponge that is inserted there. And remember, this is not a plant. Sponges are animals. And these are marine animals. Asymmetrical, they are shapeless and have cellular level of organization. No tissue formed. That's why it is kept in the first phylum. They have but very good system, water transport system or canal system. This is a clue in future that when the animals evolve, there will be circulatory system. Water enters through minute pores called ostia into central cavity called spongocoel, from where it goes out through osculum. See, these are all the technical words that you must recollect. And nutrition, respiration and excretion is performed by pathway of water transport system. So it is a multi-purpose use. Skeleton is made up of spicules, spine-like thing, thorn-like thing, and spongin fibers. Egg and sperms are produced by same organism, and then if an organism produces both the gametes, male and female, that animal is called hermaphrodite. sexual reproduction by fragmentation, and sexual reproduction by gamete formation. Fertilization is internal and development is indirect. There is no direct development, indirect development. Example, cycon, spongilla. What do you mean by fertilization internal? It is inside the organism. Development of the young one is inside the organism. And the development, when we say indirect, it is through, there is something called embryo. Okay, there will be what we call, it is not a direct, like in our case it is direct development. Mother gives birth to the young ones. Here it is not the young one, via media. Then we say it is indirect. And second, phylum Nidaria or Coelenterata. These are aquatic, mostly marine, sessile, free swimming, radially symmetrical. See, radially symmetrical. You can remember it as hydrophyllum. They exhibit a tissue level organ. There it is cellular, here it is tissue level. Diploblastic, 2 layers, ectoderm and endoderm. Coelomate, coelom is there with a single opening. They show 2 types of body called polyp and medusa. This is important. Okay, dimorphism. Polyp and medusa. Polyp is sessile, fixed and cylindrical without gonads. Example, Hydra, Adamsia. Medusa is free swimming. This you must remember here. Many times he will ask you in your NEET exam, which is free swimming, umbrella like having gonads. Is it polyp or medusa? You must remember medusa. Medusa is free swimming, umbrella like chhatri. Okay, umbrella like having gonads, like Aurelia and jellyfish. Some cnidarians exhibit both forms, obelia. Polyp produce medusa asexually and medusa produce polyp sexually. This is very important. This is the best feature of Coelenterata. Right? Going ahead, we will discuss about other things in our tomorrow's session. Until then, keep on practicing, note down the important points and draw the diagrams. Post